Imagine taking a small sample of your skin or blood and turning it into sperm or eggs. No donor, no ovaries or testes, just a lab bench, some stem cell magic, and time. That idea, in vitro gametogenesis or IVG, isn't science fiction anymore. It's moving from mouse labs to serious human research. The promise, new paths to parenthood for millions. The risk, rewriting reproduction before we truly grasp the consequences. Today, we're diving into how IVG works, what the latest breakthroughs are, who's racing to make it real, and why regulators and ethicists are scrambling to keep up. What is IVG? It's the process of creating sperm or egg cells in the lab by starting with pluripotent stem cells. Either embryonic stem cells or induced pluripotent stem cells reprogrammed from adult tissue like skin or blood. Scientists guide those stem cells through the same developmental milestones they'd experience inside a body until they become germ cells. Then sperm-like or egg-like cells, it's the core idea behind turning ordinary cells into reproductive cells. There's also a second parallel approach, somatic cell nuclear transfer. The technique behind Dolly the sheep, where a skin cell nucleus is placed into a donor egg whose own DNA has been removed, then coaxed to shed half its chromosomes like a natural egg would. This bypasses some stem cell steps and has shown progress in animal models. How far along are we? IVG has hit some amazing milestones. In 2023, a Japanese team led by Katsuhiko Hayashi made egg cells from male mouse cells. After fertilization and implantation, a small number of embryos developed into live, healthy pups. It's proof that lab-made gametes can, in principle, make mammals. The leap to humans is huge, but the biological blueprint exists. In humans, researchers are building the pieces. Teams are developing lab-grown organoids, miniature, simplified versions of tissues, to mimic ovarian and testicular environments where gametes mature. At recent fertility conferences, leading labs presented advances such as primitive sperm cells within testis organoids and human ovary organoids, key steps to help human cells on their journey. Timelines are cautious, but several experts now say a few to 10 years is plausible for tightly controlled clinical trials, only if safety checks are met. Meanwhile, at Oregon Health and Science University, researchers are pushing that nuclear transfer route, showing in mice that a skin cell nucleus can be reset inside a donor egg to behave like a real egg, potentially a shortcut for certain patients if it can be made safe in humans. Who's racing to get there? On the academic side, Osaka University and collaborators in Japan are widely seen as leaders, with multiple firsts in mouse IVG and germ cell development. In the startup world, Conception California publicly states its mission is to turn blood cells into human eggs via IVG, aiming for a proof-of-concept human egg under strict lab conditions. Reporting has linked the company with high-profile Silicon Valley investors, reflecting intense interest in moving IVG from the bench toward future clinical use. Across Europe, major societies like Escher have put IVG on center stage, while the UK's HFEA says the technology is nearing viability, but not ready for treatment, and is calling for statutory regulation tailored to IVG before any clinical rollout. Why it matters? If proven safe, IVG could open biological parenthood to people currently excluded. Cancer survivors whose fertility was lost to treatment, those with diminished ovarian reserve or non-obstructive azoospermia, same-sex couples seeking shared genetic connection to a child, people who need many eggs quickly for pre-implantation genetic testing to avoid severe inherited disease. Beyond individual families, IVG could reshape fertility care, less reliance on donors, potentially broader access, and entirely new options for preserving fertility earlier in life. But every one of these benefits must be weighed against biological risks and societal trade-offs. What are the big science challenges? Safety is the wall IVG must climb. Genetic and epigenetic errors. Lab-made germ cells must imprint the genome correctly. Even subtle mistakes can cause developmental disorders. Long-term follow-up. Proving safety means tracking outcomes across generations, not just at birth. Efficiency and quality control. Mouse success rates were low. Humans are far more complex. Leading scientists emphasize years of validation before human use. Regulators and journals echo that message. Promising science does not equal clinical readiness. Ethics and law, who should decide? Ethics boards are already flagging tough questions. Should IVG be limited to medical infertility or allowed for elective reasons? Could IVG enable multiplex parenting more than 2 genetic parents? And should that be legal? What about embryo selection at massive scale? Does it amplify inequality? The UK's HFEA currently prohibits the clinical use of IVG under existing law and recommends future-proofed regulation before any treatment use. Independent bioethics groups call for early public debate, so society, not just labs, decides the boundaries. What happens next? Watch for 3 milestones. Robust human germ cell maturation in organoids that pass gold standard genetic and imprinting tests. Regulatory frameworks that spell out when, how, and for whom IVG could be used. Transparent, peer-reviewed data, not just press releases. Showing safety in preclinical models before any human embryo creation is considered. IVG could become one of the most consequential medical technologies of our lifetime. The question isn't only can we do it, it's when we can, how should we use it? Stay curious, stay critical, and tell us where you would draw the line. How do you feel about the science and ethics of IVG? Write down your thoughts in the comments. And if you think society should or shouldn't greenlight this tech, tell us why. Like, subscribe, and share to keep this conversation going. Stay tuned for more videos to come.